Morning all. So today indeed I would like to talk to you about whether manually determined or visually biases your quantitative perivascular space measurements. And so for this talk I would also like to focus on the perivascular space quantification in CA, or especially hereditary CA. So my declarations, my PhD project is made possible by independent financial support from the TRACK DCA consortium. This is independent of this study and of these results. The CJ Horter Center receives support from Philips. So first I would like to start off with some background on CA. So CA, or cerebral amyloid angiopathy, is a cerebral small vessel disease that is characterized by arterial amyloid beta accumulation. So this is mainly in the cortical and leptomeningeal arteries of the brain. And it is one of the leading causes of intracerebral hemorrhage. It's related to multiple imaging markers that are depicted on the image on the right. So one of them is impaired vascular reactivity, and this can be measured through visually stimulated fMRI. We also see hemorrhagic markers, multiple of them. So an example is microbleeds, which you can see in the bottom here. Intracerebral hemorrhage, cortical superficial siderosis. So this is a hemosiderin deposition along the cortex, or the subarachnoid hemorrhage. There are also non-hemorrhagic markers such as microinfarcts, enlarged perivascular spaces, and white matter hyperintensity. So recently, this pathophysiological framework has been introduced by one of our colleagues, Emma Koemans. And she introduced the idea that different stages follow into each other. And so that the first stage is the amyloid beta deposition in and along the vessel walls. Then the second stage is loss of vasoreactivity, which can be measured through visually stimulated fMRI. Then the next stage, you see that the smooth muscle cells in the vessel wall, they're starting to disappear and being replaced by its amyloid beta protein. And then you get non-hemorrhagic injury, and this is related also to cognitive impairments. And so to say, the last stage is the stage of symptomatic hemorrhagic lesions. And so CA has also been related to brain clearance. So we think that in impaired brain clearance, it might play a role in CA, and it's even hypothesized as a causal factor. And so this is partly depicted on the image on the right. So you can see in the cortical part of the vessel, you see amyloid beta accumulation. So this is along the vessel wall. And then in the white matter portion of the same vessel, you see enlarged perivascular spaces. And this indicates that there is something going on, that this amyloid beta is not being cleared well enough from the brain. So we all have amyloid beta within our brains. But it's pathological if it's actually accumulating in the vessels, and then something is going wrong, but we're not really sure what is going wrong. So you have a sporadic form of CAA. This is basically an elderly disease, and it can be diagnosed through the Boston criteria. I will not go into depth too much about this, but it's partially based on intracerebral hemorrhage and white matter pathology. And sporadic CAA can only be diagnosed in vivo on a probable or possible basis, and only a definitive diagnosis can be made post-mortem, so based on histopathology. There are, however, also genetic forms, and one of these genetic forms is Dutch type CA, or from Dutch people among us also known as the kat-ziekte. It has an autosomal dominant inheritance pattern, and that means that if you have the gene or the gene mutation, you will get the disease, and the chances that you pass it on to your child are 50%. And so we can use genetic testing to confirm this diagnosis, and this also allows already to study this disease in the early disease stages. So for example, in the pre-symptomatic phase, so before one becomes symptomatic, and usually you only find these cases when someone actually presents with symptoms and goes to the clinic. So for the DCA, this is not the case. And since DCA is very similar to sporadic CA, we think that this might function as a model for CA, for sporadic CA, sorry. However, Dutch type CA or DCA has an earlier onset, and it has a faster disease progression than sporadic CA. So then zooming a bit more in on the perivascular spaces. So they're one of the markers that we see in CEA, and we think that they are related to impaired waste clearance, as has already been touched upon by Lydia. Yeah, so again, we see these enlarged perivascular spaces in the same vessels or just below the portion of the vessel in which we see amyloid beta accumulation, indicating that there is some kind of interplay between these two. There is a relationship between the accumulation of the amyloid and these enlarged perivascular spaces. Enlarged perivascular spaces have been shown both in sporadic CA, but also and in symptomatic DCA. And this has been done through single slice quantification and through visual rating. So this is an example of a patient with DCA, and there are many perivascular spaces to be seen on this image, but a few of them are indicated with arrows. So to continue on the visual rating scale, this is currently the golden standard to assess perivascular spaces in the clinic. When you do the visual rating, or when you assess the visual rating, you count the perivascular spaces on just a single slice in the centrum semio-valley and on just one hemisphere. So you select the slice where you think you see most perivascular spaces, you start counting them, and then based on that, you categorize them based on this visual rating scale. And so if one has no perivascular spaces, this person will fall in level 1. If one has more than 40 perivascular spaces on this single slice on this one hemisphere, this person will fall in level 4. So we think and we see that there is quite a strong ceiling effect because a lot of these patients actually already fall within level 4. And this is also indicated already on the picture on the right. So this is already partially some results from this study. So you see 2 patients, bottom one and top one. And so both of them fall within the 4th level of the visual rating scale, but they actually have very different levels of white matter perivascular space volume. So this white matter perivascular space volume is the volume of perivascular spaces throughout the entire brain. So not only just this slice. So for the top one, it's approximately 10 milliliters throughout the brain, but for the bottom one, it's almost 40 milliliters. But both of them would fall into the 4th category. So we think that this is a strong limitation to the visual rating scale, and this will also show limitations if you're going to do longitudinal studies. So we hope that the white matter perivascular space volume is more sensitive also to progression. So ways to assess the perivascular space volume. One of the more often used method is using the French Vesnus filter. And so this has actually been developed here in this hospital a while ago by Alejandro Aprengi. And so it's an algorithm to detect tubular structures. So it looks whether a structure is tubular, so longer than the width. And it also looks at the contrast differences within the structure and outside the structure. And based on this, you can segment these structures. And you can also apply a threshold to determine the strictness of this filter. And so this is also what this study was partly looking at. So the aim of the study first is to assess the whole brain white matter perivascular space volume in Dutch type CA. And second, we also wanted to assess the relation between this French Vesnus filter thresholds that we determined for each individual. And we wanted to check the relationship with the actual volume that we got out of this. So why do we want to do this? I already touched upon this, but we want to overcome the limitations of the visual rating. We also want to enable to assess progression within patients. We want to assess progression as is, but also accurately, of course. And this is also maybe related to a treatment trial to actually look at what happens over time with markers in the brain. And so this is also to increase understanding of mechanisms behind CA that might be related to clearance. And we also want to ensure that the visually selected thresholds does not introduce bias into our results. So then moving on to the methods, how do we do this white matter perivascular space segmentation? We first determine our region of interest. So we're interested in the white matter. So we start off with the white matter segmentation, and then we remove the cerebellum, the brainstem, the basal ganglia, and dilated CSF segmentations, since we don't expect to see perivascular spaces there, and perivascular spaces in the basal ganglia are not for CA. Then we register this region of interest to our T2 weighted space or image, since this is where we see the perivascular spaces most clearly. Here they are iso-intense to the CSF, so they are hyperintense. Then we manually adjust the region of interest, since these white matter segmentations, they're never perfect, and especially with pathology, the segmentations oftentimes fail or show limitations. So we try to adjust for that to reduce false positive PVS segmentations. Then we visually determine our Frenchy Wessonus filter thresholds. And so we do this again to avoid both positive and false positive and false negative PVS segmentations. And lastly, we calculate the white matter perivascular space volume out of the segmentation, and we normalize it for the white matter volume per. So the study sample is from the early diagnosis of amyloid angiopathy network, also known as the ADAN study. And this sample allows a comparison between different groups. So the first group is between the symptomatic and the pre-symptomatic DCA. So you fall within the symptomatic DCA if you had a symptomatic hemorrhage, and otherwise you're pre-symptomatic. We also compare between symptomatic DCA and their matched controls. Lastly, between sorry, I think I said this twice. So the second one is symptomatic versus matched controls, and the last one is pre-symptomatic versus matched controls. And so the matched controls, they are matched on both on sex and age. Then moving on to the results. So first of all, we see an increased corrected white matter perivascular space volume in the symptomatic DCA compared to their controls. So the green compared to the purple. We also see an increased volume in the symptomatic DCA compared to the pre-symptomatic DCA. And lastly, and most interestingly, we already see an increase in the pre-symptomatic DCA compared to their matched controls, so to the younger controls. And I think it's good to note here that you might have spotted this potential outlier. So we also redid the analysis without this potential outlier, and then we still saw a significant difference between the pre-symptomatic DCA and their matched controls. So this slide shows you an example of this perivascular space segmentation. So on the left, you see an example symptomatic DCA patient, and on the right, you see a matched control. And you can see when we zoom in, you see more perivascular spaces in the symptomatic case. But the bottom image also shows a rendered image for the whole brain. So you can see that this is not only the case at the level of the centrum semiorali, but actually throughout the entire brain. Then I have a similar example for the pre-symptomatic DCA compared to their matched controls or a matched control. And you can again see that there are a lot more perivascular spaces in the pre-symptomatic case with a much higher volume. And this is again completely throughout the brain and not only in the centrum semiorali. So then we also looked at the visually determined Frenchy Wessonus filter thresholds. So this graph shows the volume per participant with the visually determined thresholds. So every dot is one participant, and with the four different groups. And you can see here that the visually determined thresholds, they don't seem to introduce a bias in the volumes that we get out of it. Then moving on to the discussion and conclusion. So first, the limitations. So the current method is quite time consuming as it takes a lot of manual effort to adjust these region of interest. And the results that you get or how well the method works is dependent on your scan quality. And moving on to the main findings. So first of all, we introduce a method to avoid the ceiling effect that you see in the normal perivascular space assessments. And we measure whole brain white matter perivascular space volume. I think that this is of importance for longitudinal studies. Then the results confirm previously shown results of enlarged perivascular spaces in the symptomatic DCA. And for the first time, we also prove increased or show increased perivascular space volume already in the pre-symptomatic phase. And this may indicate that this whole brain white matter perivascular space volume might function as an early marker. And lastly, we see no clear effect of bias based on the visually determined branchiovascular filter thresholds. So that's it for my talk. I would like to thank the participants, colleagues, and co-authors, of course, and of course you all for